Hello, Shikar. All of a sudden, the network part tripped. Inverter also tripped. I don't know how. Okay, so I was discussing about this one, right? The Negro one. So a Negro, suppose a Negro person has a specific color or too much melanin synthesis due to the effect of two genes, A and B, and both are suppose are in homozygous dominant stage. Now this woman, okay, marries a man, okay, she marries a man who is albino, means no melanin whatsoever. Albino means no melanin. So small a, small a, small b, that is no melanin. Okay, albino. So now what you will be having a mulatto genotype because the gametes that you are getting is AB here, small b here. So F F not generation. So in F1 you will be getting this case. Okay. So what you will be having is this is known as mulatto. Okay. He will be white or he will be white but not completely dark. Okay, so because the quantification is not proper, means you need both the genes to be completely expressive, like what I told you in case of incomplete dominance. Okay, pink, the case of the snapdragon plant or the snapdragon flower, where the reddishness was not being expressed. Why? Because both the genes were not, you can say, both the genes which will be produced, both the alleles, one was capital R, one was small r, so this was not coding for that pigment, okay, or that enzyme which will be converting the substance white into red. Okay, so this was not converting, this was only coding for that enzyme which would convert the substance X, which is white, to red. So what was happening is half the time from this specific gene mRNA would be produced and that would make a useless protein, okay, which will not be of any use. Same way here. Okay, so that is known as quantification effect or quantitative genetics. Okay, so just giving you a treat of quantitative genetics. So always in our system, how much protein has been produced, that is of a great relevance, significance, right? So only not that it's produced by the one of the dominant genes, so you will be completely dominant. No, that is Mendelian. But when non-Mendelian genetics we study, Mendel was not right all the time because I told you he couldn't explain linkage, he couldn't explain incomplete dominance, he could not explain codominance, he could only explain the extremes of the phenotype, either recessive or dominant. That's all. The midway phenotype, which was the blending pattern, he was not able to, you can say, comprehend, decipher, neither explain those specific phenomena, right? So that is how it proceeded for genetics. Now coming on to, if you just check the cross, okay, of AB, if you just check the cross, I'll just make it. So those things are not very difficult. If you just check the cross, what will you get is, suppose F2 I am drawing, okay, just giving you a glimpse of how it could be. It's quite easy if you understand it. Small v, small a, capital V. This side you can make the same arrangement that I had shown you by that trick few classes ago, right? Now let's go for this. Small a, capital B, capital V, capital V, small a. Okay. I'll just show you how I'm getting the difference in the cross. That's all. This is the ratios that we will be getting after this specific event. One will be your albino, complete. Okay. Now wherever capital A, capital A, capital B comes, okay. So one will be your Negro, only one. You can see it's not 9331 ratio. Okay. One is your Negro. Okay. Both capital A means high concentration. Okay. One capital B, small b. That means not Negro, but still dark in color. You can call them dark in color. Okay. Again here both are capital, but this is not completely capital, means little bit lesser concentration of the protein, or the enzyme again not Negro but dark in color. Okay. Capital A, capital B, this one. Both heterozygous, means both are not able to produce properly, so whitish, so mulatto. Mulatto are the whiter ones. Okay. Now capital A, small b, not at all able to make this one. Okay, this is also heterozygous, so they will be very very fair. Okay, really fair. Okay, then again mulatto here. Then capital A, small b. Okay. That is the second part. Again there will be mulatto, because one is completely dominant. Okay, so not completely fair. Fair will be, one should be completely heterozygous, or and one should be completely homozygous recessive. Okay, here again dark, because both are not completely dominant, homozygous, right? Again dark. Okay, again mulatto, this one again fitting this stage, both heterozygous. Here one heterozygous, one homozygous recessive, so this one again fair. Okay, again mulatto M, again this one fair, fitting the same. Okay, again this one fair. Okay, so what kind of ratio are you getting? Okay, the ratio I think you are getting is 1 is to, how many dark are you getting? 1 2 3 4. 1 is to 4. Then how many mulatto you are getting? 1 2 3 4 5 6. 1 is to 4 is to 6. Then again fair. Okay, 1 fair, 2 fair, 3 fair, 4 fair. 4 and 1 you are getting albino. So that was the thing I told you, with two genes going for, you can say, polygenic inheritance. If two genes are there, A and B, you will get this ratio. So see how you are getting this ratio. Same crossing, but just interpretation way of phenotype is different. Okay, so you can count again. 6 plus 4 10, plus 4 14, 115, 116. There you are getting 9331. Okay, same 16 only, but the difference in the ratio, just see here. So that is what I wanted you to see, because one just says the ratio is different, you feel it's quite difficult, but when you draw it, okay, it is quite easy. So in entrance exams later on, if you take AP in case of biology and all, so these will be there, but these are very easy topics. So they make it sound tough, but once you have the concept in your head completely crystal clear, then it's quite easy. Okay. Do you have any doubt in this cross, Jessica? Just tell me if you have any kind of doubt here. Any queries? So, any doubts, Jessica, here in this cross? So now we'll be coming on to the next topic, which is known as epistasis. Okay. So epistasis is nothing but it's a Greek word which means standing over. Okay, it was first used by a scientist known as Bateson, okay, to describe a masking effect. Masking effect means it is a completely different phenomenon. What it means it's an interaction between an interaction between a pair of loci, means a pair of different genetic locations, you can say pair of loci in which the phenotypic effect of one locus of one locus masks, you can say, the or one locus, you can say, depends on the genotype of the second locus. That will be better. Depends on the genotype of the second locus. So this is a completely different phenomenon which has been given at the end of your chapter epistasis. Okay, means that this is an interaction between two different genes, not the same alleles. Suppose you have in chromosome number 1 here. Okay, just to give you the perspective, this is chromosome number 1. Okay, this is the homologue of chromosome number 1. This is coming from the father, this is coming from the mother. Fine. This is chromosome number 5. This is coming from father, this is also 5 coming from mother, this is 1. Now suppose you have a gene here, which are alleles. These two are alleles. But suppose the expression of these two alleles are being controlled by this one. Okay, are being controlled by the expression of these genes or these alleles or this specific gene. That is known as epistasis. Okay, that is known as epistasis. So the genes whose phenotype are expressed, suppose this one is being expressed, okay, that is known as epistatic gene. And suppose this expression suppress the expression of this one, of this gene's phenotype, so this gene will be known as suppressed or hypostatic gene. Or hypostatic gene. I'll explain it to you how does it happen, because it may sound perplexing, because this is the only part which has been given in your book. They haven't explained it. Okay, so I'll explain it to you so that the concept gets in your head. So this is how epistasis works. It's not that this allele is dominant over this allele, not like that. Here this entire gene is dominant over another gene. These two genes are completely different. These are homologous chromosomes, these are alleles. But the gene, this one and gene 2 are not alleles. They're completely different set of genes, different set of alleles. But one set of genes or one set of alleles, one gene is able to mask the or effect, or you can say adversely affect the phenotype of the other gene. Okay, which are at completely different positions. That is why they are known as loci. That is the plural of locus. Locus means where do you reside, which is your present, you can say, location, position. So there are two different loci. Loci 1, its loci is at chromosome number 1. Sorry, its locus is at chromosome number 5. Okay, so its locus is here, chromosome number 1. So both are at different loci. The loci is different. So this is an intergenic, you can say, relationship, not intragenic where you are just looking at the relationship between the two alleles. This is a completely different intergenic relationship. Okay, that is known as the you can say fundamental concept behind epistasis. Okay, so now I'll tell you how does epistasis happen. Okay, so when you talk about, you can say, recessive epistasis. Okay, there are many types of epistasis. Let's talk about, first of all, I'll give you a certain example. Okay, the way you will be able to comprehend it in a much easier way. Okay, there are multiple types of epistasis happening. Okay, you have, you can say, dominant epistasis, recessive epistasis. You have duplicate recessive genes. Okay, you have duplicate dominant genes. Okay, so there are multiple ways of epistasis happening. But the thing is, let's talk about a simple example which we had talked about in the blood group stage. Okay, like we had talked about the blood group. So I told you to the last galactose. Okay, to the last galactose, what I told you is you have something attached known as FUT gene. Okay, FUT1 gene, you have something known as fucose attached to the last galactose. If you remember it properly, I'll just show it to you also. So I'll draw that diagram again. This was, you can say, ceramide. Then we have, you can say, glucose, then galactose, and then we have N-acetyl galactosamine, then again galactose. Directly change the color. This is N-acetyl, you can see. And then we have again here galactose. This is, and to this one you have attached fucose. Okay. The gene here is FUT1, which will produce this fucosyl transferase enzyme that attaches this to fucose. Now this is the complete H substance. Now understand it properly. Suppose, okay, you have something known as this phenotype IAIA. Okay, means that to this last galactose you will be able to attach which one? One more, you can say, N-acetyl galactosamine. Okay, one more N-acetyl galactosamine. That is fine, attached. And suppose another way is, you have another phenotype. Suppose you have both IB, IB also. So here what you can do is ceramide, glucose, galactose, then N-acetyl galactosamine. Then you have one more galactose. To that I can add one more galactose. Here I can add glucose. Okay, this is, you can say, blood group B. Now this is blood group A. Okay, suppose you have both the alleles, or a person has A allele, a person has B. One person has blood group A, person number 1. Person number 2 has blood group B. You know, whatever the way it suits you for better understanding. Let's suppose one condition is, next condition what I am doing is, a person is recessive for this specific gene, means it doesn't produce FUT1 gene proper, means it doesn't have proper FUT1 gene, it has some kind of missense mutation or nonsense mutation which put the stop codons in it, and therefore fucosyl transferase is not produced. Okay, Either not produced or it is completely dysfunctional, of no use or dysfunctional. So what I mean to say is, suppose fute gene is on chromosome number 1. Okay, fute 1. So both the alleles are gone case here. Okay. And suppose this A gene is on chromosome number 5. Okay. This is chromosome number 5. So you have IA, both dominant, you have proper. So what will you expect, Ishika? You will think that because it has proper IA, the blood group will be A, right? But the thing is the recessiveness of this one, the small f, the recessiveness of this gene will do what? Will not add this fucose to the galactose, last galactose of the H substance. So the H substance will be incomplete. What you will generate is an incomplete H substance. And to the incomplete H substance, the N-acetyl glucosyl, or sorry, galactosyl transferase cannot recognize this incomplete H substance. In order to recognize this substrate, it needs this fucose to be present. If this fucose is not present, even if this enzyme is present, which will add N-acetyl galactosamine to the last galactose, cannot add N-acetyl galactosamine to the last galactose because it cannot recognize this incomplete S substance. For S substance to be completely complete, it needs fucose to be attached to the last galactose. Okay, then N-acetyl galactosamine or one more galactose can be attached. Okay, so what happens is, suppose you have a genotype small f, and that side you have capital this one or both. What is happening? The genes at one locus is suppressing or masking the effect of another gene. So this is the epistatic gene. This is the hypostatic gene. Even though it is dominant, it is of no use. Right or wrong? This is the molecular reason of how this happens. Every book will show you the genetic reason it happened, but you will always think how recessive here, dominant there, how it is able to mask going by different chromosomes, how it is able to mask the gene of different chromosomes. You will think that that chromosome is completely different. It has dominant, so it will express. What is the problem? Why will I not see or observe the phenotype? This is why you won't observe. This is the molecular reason which none of the books explain. And that's why students find this epistasis to be extremely difficult because they are not aware of the molecular reason behind epistasis. So this is the molecular reason behind recessive epistasis. Means epistasis by a set of recessive genes at one chromosome is able to suppress the set of another gene which are dominant on the present on some other chromosome, human blood group H substance. And this thing is also known as Bombay blood group. Very, you can say, rare blood group known as Bombay phenotype. In Bombay blood group, what happens is it looks like O because A and B, even if you have the genes, you cannot attach. So what happens is, if you look like O, that you are not even O. O will have complete S substance, but here you are lacking fucose. So it is not even O. So if you lose blood and you are transfused with blood group O, still you will be able to reject it. Okay, because it is not even O. It is an incomplete H substance. O is a complete H substance with fucose. It is an incomplete H substance. This specific, very rare case, rare phenotype known as Bombay blood group. A lady in the city of Mumbai, which is still, you can say, known, or you can say formerly known as, its previous name was Bombay. So she was present in that city, and the first time the scientists made sure or were able to discern that when she had a bleeding episode. Okay, Bombay blood grouping. So that is how it completely undergoes. Okay, Bombay blood group. Okay, now tell me, Ishika, do you have any problem in today's lecture? Epistasis I have completed. So for the next class, I am only leaving genetic linkage and recombination. So I hope linkage and recombination along with meiosis we will finish in I'll give you the aspects of meiosis, and also we'll finish linkage and recombination in a very, very conceptual manner. I'll tell you some shortcut points also by which questions you will be able to solve with ease regarding linkage and recombination. Thomas Hunt Morgan, who is known as the father of the you can say modern genetics. The father of the classical genetics is Gregor Johann Mendel, whereas T.H. Morgan and his experiment using Drosophila melanogaster, one of the best genetic tools. Okay, why is it the best? I'll also explain to you why Drosophila is chosen. The common name is the fruit fly. Okay, okay then, Ishika, enjoy your day. Have a nice day. Thanks a lot. Let's meet you in the next class.